Good morning. This is a re-recording of a presentation given in Bovino, Italy at the International Cannabinoid Research Society on July 1st, 2014. This presentation generated a tremendous amount of interest that I felt obligated to give it another recording and address some of those questions for the audience at large. This is very much a serendipitous story. It's a story about a patient who is on a diagnostic odyssey that's not unlike this odyssey in the movie or the novel Life of Pi, where a gentleman stuck in a boat with a hungry Bengal tiger and miraculously survives this odyssey. Well, this odyssey here is about a patient who has EKGs that look like this. And the one there in the upper right is actually ventricular tachycardia, which can in many ways be just like Mr. Parker or Bengal tiger and strike sudden death at any time. So the story actually dates back to a startup company, the name of Cortigen Life Sciences. And this was a founder of the company who was going through rapid growth in the company, almost 100% year-over-year growth, which led to a fairly stressful environment. And he came down with atrial fibrillation. This atrial fibrillation became its own little diagnostic odyssey. But fortunately enough, and serendipitously enough, the company that he had founded was building next-generation sequencing tools to try and unravel diagnostic odysseys. But these tools are mostly focused on mitochondrial dysfunction, which plays a part in epilepsy and in developmental delay in autism, and there is some overlap with cardiac disease, but it's not been the company's core focus. I think the best overlap there exists today is with the epilepsy sequencing work that they're doing. This is a project that sequences 500 genes related to epilepsy. These genes are very important in guiding therapeutic management of patients with complex epilepsy. So there's 37 different genetic indications for epilepsy. There are cases where people will have sodium channel mutations like SCN1A, and you need to avoid sodium channel blockers with these types of mutations. And interestingly enough today, and very relevant to this community, is that 30 to 40% of these SCN1A patients respond to cannabidiol or CBD. There are also GLUT1 variants in these patients that sometimes can lead to a positive or be predictive of a positive response to the ketogenic diet. And there are POLG variants that you need to avoid valproic acid, otherwise you'll have fatal liver failure. And there are some GABA receptor contraindications with steripental and some of the P450 genes that are involved, and this test does in fact sequence all 24 of the P450 cytochromes. And there's a handful of other contraindications that may help inform decisions, like absence seizures and Dravet's have some calcium channel variants that are collected to them. I think the most striking one, which is not yet in the panel but being built into it today, is HLA 1502. If you have that variant, which is 20% in the Asian population, you will respond very negatively to carbamazepine, which can in fact give you Stevens-Johnson syndrome, which has a 30% mortality rate. So in sequencing these patients, we recognize that a good number of these genes in fact overlap with the genes known to be involved in cardiac disease. And so when this patient wasn't responding to conventional therapies and was left out on his own, we decided to deploy what we knew how to do best at Cortigen and sequence the patient. So a little bit about that patient. Okay, this is a 40-year-old who presents with a new onset AFib and rapid ventricular response, 180 to 190 beats per minute. This is a very healthy individual, a vegetarian, second-degree black belt, no structural heart disease, and has a prior history of some palpitations throughout the family. In this particular case, the patient was triggered by ethanol, caffeine, and stress. These seem to be three critical ingredients to any startup environment, so no surprise there. But the other familiar history here is that the father has AFib that has been responsive to flecainide, which is a sodium channel blocker, but does have affinity for the ranitidine receptors, which are calcium channels. And there is some stroke on the maternal line with palpitations. So there may be both maternal and paternal complications with this. So when the patient would go to the ER, serendipity would have it after waiting in the ER to be hooked up to EKGs. He'd eventually spontaneously convert to sinus rhythm, and it would evade detection. So this is where these very handy AliveCor systems, which are iPhone-based EKGs, come into play, because this mobile technology, I really think, is bringing on a great convergence in medicine, where we have mobile technologies, we have genomics, and we have the great promise of the endocannabinoid system all coming together to help unravel these these personalized medicine diagnostic odyssey stories. But in this case, once we're able to capture better EKGs, a physician could go about thinking about a therapeutic strategy, whether it be calcium channel blockers or beta blockers. Now, these EKGs did show a few signs of what may be VTAC, and that is what really drove the urgency in this case to try and take a more extreme approach. When given calcium channel blockers or beta blockers, they induced bradycardia, and the patient couldn't get out of bed. Heart rates of 30 or less. So at this point, he was stuck considering an ablation or more experimental therapies. Now, a little bit about AF. AF is a fairly common disease. It's 3 in 6 million people in America affected by it. It's expected to double the next 25 years, and it's expensive, and it can add up to $9,000 a year in costs, or $26 billion to the U.S. healthcare industry. So at this point, facing ablations that might be difficult to map, considering the diversity in EKGs that the cardiologists were seeing, we stopped to consider exome sequencing. Now, we started sequencing with the Cortigen panels, and we found some of the variants of the panels, but we went on to exome sequencing to ensure that we didn't miss any other cardiac genes. And I'll explain that in just a minute. But both methods rely on the same technique, which is using molecular inversion probes to go and capture 200,000 different exons in the genome. We then amplify those with a method we published in October called Dream PCR. And this is a method that helps navigate many of the gene patents that still persist in the United States, but it also is very instrumental in eliminating any contamination, which in course allows and enables better mosaicism detection, which can be very important in many neurological sequencing projects. So once we have these PCR products, we put them on these glass chips from an Illumina MySeq system, generate millions and millions of clusters on these glass slides, which we then sequence and then turn into 50 to 100 million reads in a matter of 36 hours. Those reads are aligned to the genome, and we can then begin calling variants. Now, the variant calling process has been fairly streamlined and standardized and is all in the cloud today. The real challenging part that isn't really cloud-enabled yet is that the clinical interpretation of those variants often requires MDs, variant scientists, and some genetic counselors, and this is very much still an art today. So a bit about clinical sensitivity here. Although exome sequencing is quite popular in making the news in New England Journal of Medicine, there are some limitations you need to be aware of. They are an inch thin and a mile wide. They generally produce 92% coverage of the coding content, which sounds like an A in school, but it's an F in genomics. This is 1 in 12 bases missing. Some of these are creeping up to 96 or 95% coverage of the coding content, over 20x coverage, and that's probably the unpublished work that's really just emerging today. So these numbers are, of course, increasing over time, but as matters stand today, this does have some negative consequences for picking up more complex variants in the genome, like insertions and deletions. These can be far more difficult to pick up at 20x coverage, and you only pick up 67% of those. This is important. This is very important for clinical sensitivity, because if you look at the size of variants that are in a human genome, and if you look at this in ClinVar or HDMD, you'll see that there is a log K to scale, decay in a scale of the number of variants there are of different size range. So SNPs are single base pair variants, and then there are 2, 3, 4 base pair insertions and deletions that occur, and they occur in increasingly higher frequency the smaller they are. So it's important to be able to pick those up, and that requires high depth and comprehensive coverage. To get that, what people often do, and what we do at Cortigen, is resort to gene panels where we focus all the sequencing horsepower and the coverage over the genes we know are in the clinical hypothesis. And these are called gene panels. So the EpiSeq product we sell has a 500 gene panel, which gets us closer to 98% of the areas covered and better indel sensitivity. We combine these two. Whenever there are negative EpiSeq results, we will often reflex to exomes, and they work hand in hand quite nicely. Now, exomes produce a lot of data. We get 101 meg of target selected, so 2/3 of the sequence that we target is actually intronic, and that's so that we can get better capture of the splice junctions. Typically, you're sequencing these to 80x mean coverage, and the gist of this slide is that it generates lots of variants, and we tend to filter those variants fairly aggressively to bring us down to 1,000 amino acid changing variants. The filtration is based on population frequency today, and in addition to population frequency, we will try to deploy some clustering of these variants in genes that are in similar pathways. This can be very handy in finding where the mutational burden is, particularly in channelopathies. Channelopathies are known to be hyperpolymorphic genes, and so it's very easy to draw false conclusions and type 1 errors in this type of setting. So this led us down to 58 cardiac-related genes that were of interest and had variants in them, but we noticed that there were 2 of them that were intimately related that had 5 different variants in them, and that kind of stood out to us as being something that we should pay more attention to. So these were the ryanodine receptor 2 and CACNA1C, often known as calcium V1.2. It is an L-type voltage sensitive calcium channel, and we'll touch on that in a little bit. Before we go there, let's look at ryanodine receptors. So RYR2 is a dominant gene. Now, that means when you have a heterozygous base, you pay attention. I think what we're going to find in the next 2 years is this concept of a dominant and recessive gene is going to go away, and it's going to come down to a residue resolution, all the way down to the amino acid. You can have differences into what's dominant and recessive just based on where the variant is in the gene. But today, we still take notice when a dominant gene is hit, and we call heterozygous variants there more aggressively. Now, the next thing you do if you know it's in a dominant gene is you then see if the gene or the amino acid is conserved, and is it conserved through evolution. In this case, we look at the evolution conservation across 44 different species, and this one's conserved all the way back to lamprey, which is a blood-sucking eel. And that's very far back, almost 500 million years in evolution, so Darwin does not want this residue to change. We also use some protein prediction tools. SIFT and polyphen are probably 2 of the most common, and they both call this a damaging mutation with the highest score it can generate. But the reason we don't just use these 2, we use actually 5 different tools to do this, is because these things are imperfect. They're trying to make 3-dimensional information out of 2-dimensional sequence data, and there's just so many limitations with that you can see these false positive rates on the upper left graph here versus the true positive rates leave a lot to be desired. So what you really need to do, and what we like to do when possible, is to dive into the 3D structure of the protein. Here's a beautiful picture of the ryanodine receptor, where you can see this pore-like structure that when other calcium is present on the external face of this pore, it rotates it open so that calcium can then flood out of the sarc into the intracellular space. So it sits on the sarc, and it is a calcium-induced calcium response, which means when the intracellular calcium gets to a certain threshold level, that pore rotates open and it floods calcium into the intracellular space to drive contraction. Okay, keep your eye on this L-type receptor over there on the left, right in front of it. That's going to become pertinent later. So, where in the protein is this? Well, this is a graph of all the polymorphisms that have been seen before, some of which have been related to cardiac disease, some which have not, that are in the gray areas. You can see the hyperpolymorphic regions of the receptor in gray. This variant sits on a highly conserved area. These horizontal areas, it's probably the most horizontal area of the receptor, is right where this one falls. And it's a proline change. This is a proline to serine. Now, prolines are like right angle brackets in peptides. When they change, we pay attention. And it happens to be changing right in this extracellular space at the center point of where this pore is rotating. And so this is by no means a smoking gun, but it certainly is raising our interest in what this variant might do. So next we go and we look through all the variants we've seen in our databases. Now, we sequence mostly pediatric patients, and this is an adult onset AFib that many people would assume has very little genetic strength to it just because of late onset. So keep that in mind here. But CVS patients are cyclic vomiting patients, and we do get lots of hits in this gene for these patients. These patients often exhibit POTS, which is a form of tachycardia. So there is some overlap there, but none of them show the exact variant. And there are some cases of negative controls, patients which aren't sick yet, that are in green there, that have hits to this receptor as well. So, there's only so much we can go with these database searches. We really need to step forward and look at how well this segregates in the phenotype of the family. So we sequenced the rest of the family, and the sister's the least symptomatic, and the 2 other brothers are showing some signs of palpitations, but nothing captured on EKG that would imply arrhythmias or VTAC. And there was also a second variant in this gene that I didn't spend much time on because it was in 14% of the population. It could perhaps be contributing in some polygenic way, but the sister's homozygous for it, and she's the least symptomatic, and so this is one that we've kind of pushed aside. Okay, so on to the other three variants. The other three variants sit in that receptor I was showing you earlier, which is the calcium receptor that's responsible for pumping calcium into the cell. And so this is a voltage-gated channel. So when the voltage, there's a voltage differential, it opens and drives calcium in. Now, there are some interesting subunits on this. I'm going to point your attention to these alpha subunits. We're going to get to those in a little bit. But there's a lot of literature links to hits in this gene causing Timothy syndrome or Brugada syndrome. Ranitidine tends to be more CPVT hits in the literature. So slightly different form of arrhythmia that is seen with ranitidine hits than with calcium hits. I've not seen any publication showing patients which have hits in both. Interestingly enough, this receptor is, or channel I should call it, is very intimately linked with the ranitidine receptor. It has its own RYR2 interaction site right there. And so that makes us pay a particular attention to variants that are in both of these genes. So how does this cluster in the family? Well, very similar story here. The brother and his most symptomatic brother are homozygous for all three. Now, two of these are fairly common, 33 and 24% in the population. There's a private variant in the family that's homozygous, surprisingly, but it is not actually 0. It's 0.016 in the ESP 6500. So there is some frequency in the population, which is very, very low, and this family tends to have homozygosity for both. So which one of these variants is to be concerned about? Well, they're all pretty close together, is one thing you'll notice. And one of them is methionine, which is something to pay attention to as we move into this slide. This is exon 43 and 45, and this maps to a very similar region in this paper that suggests there's an internal exonic promoter in this area, which is driving the expression of a C-terminal peptide that's called the CCAT region. It's part of the voltage sensor that interacts with this channel. And this region, this internal exonic promoter, actually is known to make a non-coding RNA that regulates the expression of that CCAT. So we've got a methionine change in there. We've got three variants in there. Protein prediction tools aren't going to predict this. This is something that we have to keep on our radar as we drive a hypothesis forward. So driving a hypothesis forward is still very difficult. We know we've got broken parts in here. We don't know how they're broken. We don't know which direction they're broken. All we know is that they could both be leaking this direction, or we could have them leaking in opposite directions, or we could have them included in one direction and leaking the other direction, and vice versa. So 4 different hypotheses, and perhaps there is no surprise here that calcium channel blockers aren't working. These calcium channel blockers have a variety of different classes of calcium channel blockers, but invariably they hit multiple calcium channels, and so perhaps this is creating an undesired effect of bradycardia. So at this point we thought, well, what if we can drive calcium homeostasis? That might be the better path to drug this, and we began thinking about the mitochondria, because the mitochondria are so critical in calcium homeostasis. And I was excited and ecstatic to see, and literally jumped out of my chair when I saw this work from Duncan Ryan out of Roger Pertwee's lab. This demonstrated exactly what we're looking for, which was a compound that could regulate intracellular calcium levels, and lo and behold, in the mitochondria. And this compound happened to be a compound we're very familiar with from the epilepsy space. It's called Cannabidiol. It's happening to have a very productive role in treating Dravet patients. We don't really know why yet, but maybe it has something to do with calcium. So we were a little worried this was some confirmation bias because we are so intimately involved in CBD and epilepsy sequencing. It seems as if we're trying to solve all problems with one hammer. So we reached out to many people in the field to try and get some peer review on this, an informal peer review that is, just verbal, and they forwarded us many papers. And we're very thankful for those. I'll mention those individuals at the end, but many of these papers came forward showing this is not such a crazy hypothesis. There's cannabidiol working in hypoxia ischemia in cardiac arrhythmias. We have other links to cardiac dysfunction with cannabidiol, to PPR gamma. We also have evidence that CBD increases internal anandamide levels, and that serum anandamide levels going up can actually be antiarrhythmic. And we've got direct links to VDAC1 in the mitochondria, which would make a lot of sense here. And we also have evidence that ryanodine receptors regulate endogenous cannabinoid mobilization. And so this pathway is certainly in communication with where CBD, one of the targets of CBD. So, with that said, the patient was then considering ablations versus experimental therapies, and having seen the lack of toxicity with CBD and the fear seen in the literature of sudden death with many of these types of receptor hits, the patient opted to source this through medical marijuana states here in the United States. And these laws seem to be getting more and more in favor of CBD use. Our only concern was that we measured the THC levels. There have been some signs of THC inducing tachycardia, and that's probably not something we're looking forward to here. And so we ran some TLC and asked for some better QC performed on these products before use. And the patient then titrated this up to 25 mgs 3 times daily, and we have sinus rhythm. Oral mucosal administration in an oil, and I think that's very important. The bioavailability is going to be different if it's a purified compound versus whether it's coming from the plant, and many of these oils have high amounts of omega-3 and omega-6s, which can alter the endogenous cannabinoid synthesis, so it's important we understand what these are in. Some of these were offering CBD in peppermint, which we avoided. Peppermint has been shown to be pro-arrhythmic, so I think we've got to be very careful in singing the praises of the story in terms of what type of CBD it is and what the background cannabinoids are and what it has been solubilized in. So this is a great story. The patient's ecstatic that this is working. Now, this is an N of 1. There's not a great control. The only control we've got going forward on this is that once the CNN story came out on this fabulous work with Paige Fiji and Dravet syndrome, CBD became very difficult to get, and he went through 3 droughts, and in each one of these supply droughts, his arrhythmias returned. So patients been about AFib free for 10 months, with the exception of those 3 droughts. There were frequent hospitalizations beforehand. He's led him bedridden and out of work for multiple days, and there was fear of him actually getting in a car. So I think the only testament to the story that is worth its weight is the patient has gone on to get his 3rd-degree black belt while being on CBD. So clearly his heart rhythm is in check, or at least back to where he wants it to be. But we have lots of caveats. This is an N of 1 story. This doesn't imply CBD use for all rhythm problems. And these variants may just be private to this individual and not manifest themselves in other arrhythmia cases. And so how do we prove the model? Well, what we're looking to do is generate iPS cells from the family. And using some of these new heart-on-a-chip technologies, we can split those cells and then take one set of them in CRISPR and Cas9, modify them back to wild type. And once we have those 2 populations of cells, we can test the theory of these changes with the entire genetic background of the family present. We can then apply drug and ethanol and other stressors to try to see if we can recapitulate the rhythms. And this has been shown to be a very productive model with some of the work down here in George Church's lab and Zheng Wang using heart-on-a-chip. We think we can replicate that in this model. Now, a little bit about our support here. We are privately funded. We have just applied for the first of a few of our grants. We have received an announcement of one of them, but most of this work is from 120 investors who have privately funded our efforts. If you're interested in supporting any of the IPS work, we have put together a Bitcoin address up here where we will transparently deploy those resources to test out the IPS cell model. We have another program in the company that spends some time trying to understand the genetics of the cannabis plant. There's an iPad app that supports this. For every 500 downloads of this app, we commit money to sequencing one more strain. The most recent strain that was sequenced and put forward was Dr. Grinspoon. And we do want to thank Dr. Grinspoon in that regard, in that he provided us connections to Raphael Mechoulam to get more literature on this arrhythmia work. And we also brought Raymond Brigada in here and wanted to thank him for his feedback on whether this was a likely hypothesis or not. And Rick Bowles, who, of course, was the first person to point out the severity of these ryanodine receptor hits. And I hope this brings the story full circle to the Life of Pi story. You can recognize how serendipitous this, in fact, is, that this patient happened to be in an environment that had this type of support. So with that, I'll leave it open to questions, and thank you for your time.